Good morning, good afternoon, good evening. Welcome back to Chapter 16. We're talking about domain Eubacteria. Remember, there are 3 domains of life. All living things will fit into one of these 3 categories. They either fit into domain Eubacteria that we're talking about right now, domain Archaea that we'll talk about in a bit, or domain Eukarya, which is if we went around the room and asked everyone to name a living thing. Unless you named a bacterial species, everything we would name would be in domain Eukarya. A tree, a shrub, a blade of grass, a great white shark, a blue whale, a chanterelle mushroom. All of those things are in this 3rd domain called Eukarya. Right now we're in domain Eubacteria. Bacteria reproduce by binary fission. We talked about this in Biology 1. Remember, in Biology 1, your cells will reproduce through mitosis. Prophase, metaphase, anaphase, telophase, all those complicated procedures, breaking in half. That's not how bacteria do it. The way bacteria do it is way simpler. It still is one cell that divides into 2, but there's no big stages like prophase, metaphase, anaphase. Anyway, binary fission. What does binary mean? Well, what does bi mean? A bicycle has 2 wheels. Biannually means twice a year, so bi means 2. Binary fission means to break or to split. So binary fission means literally to split in 2. They split in 2. Bacteria can reproduce at a rate of about once every 20 minutes, which means within an hour they can have doubled 3 times. So if at noon I had 10 bacteria in my leftover plate, by 12:20 there's 20 bacteria in my leftover plate. By 12:40 there are 40 bacteria in my leftover plate, and by 1 o'clock there are 80 bacteria in my leftover plate. So bacteria under the right conditions can reproduce every 20 minutes. This is why it's very important that once your leftovers have cooled to close to room temperature, stick them in the fridge, because that will drastically reduce their ability to reproduce at that 20-minute interval. 20-minute generation time. Reproduce every 20 minutes under ideal conditions. It's very common for bacteria to randomly mutate. Mutate means a random change in the DNA, a random change in the genetic code. Please remember, most of the time mutations are bad. They are lethal. They kill whatever organism the mutation is in. But remember, I said all the time? Most of the time. Which means some of the time a mutation can be good. Well, if you've got a 20-minute generation time, that means you can generate mutations really quickly. Bacteria are really good at adapting to their surroundings. Very, very easy for them to do that, which is why we've got so many species of bacteria that are antibiotic resistant. And again, random mutations will increase their genetic variation. Genetic variation is what every population needs to be able to survive in a changing environment. If the environment changes, you need to have lots of variation in your population, so some of the individuals hopefully will be able to resist this ever-changing environment. We said a little while ago in the previous video when we looked at that diagram of a bacteria, that bacteria have 1 chromosome, 1 circular chromosome, and then we also pointed out that they can sometimes have a smaller chromosome referred to as a plasmid. A bacterial chromosome holds about 2,000 to 5,000 genes. It really just kind of depends on the bacterial species. A plasmid may only have 1 gene on it. A plasmid may have 10 or 20 genes on it. So a bacterial chromosome, a few thousand genes. A plasmid, a few genes. Much smaller, but it's still circular. This is a bacterial cell right here that has spewed out its DNA, that has spewed out its chromosome. So again, look at this on PowerPoint rather than on the screen. And all these little strings, all these little lines that we see, this is that one circular chromosome. Just imagine, if you took a string of yarn and you wrapped it all the way around your house and then tied it into a circle, all right? That's going to be a huge circle. Well, that's what a bacterial chromosome is like. It's relative to the size of the bacterium, it's really big. All right, so this is that bacterial chromosome. You may or may not be able to see it in the video, but there are a couple plasmids here. All right, the plasmids, if the chromosome is the size of a loop of yarn that goes around your house, then the plasmid is like the size of a rubber band. A lot smaller. A lot smaller. And there are a few plasmids that we see that have been spewed out of this bacterial cell. There are a few different ways in which bacteria can make food. The not primary way. The first way that bacteria can make food is by being a photoautotroph. Photo, it means what? It means light. Auto means self. Troph. What does troph look or sound like? Well, troph looks or sounds like trough. And what is a trough? Like for feeding animals. So trough maybe means to feed or to eat. So photoautotroph, light, self, feeder. These are things that use light for energy. They use carbon dioxide as their carbon source, and they're able to make food out of it. Sunlight is the energy source, CO2 is the carbon source, and they're able to use that plus water to make sugar, to make food. Photoautotroph. There are a few species of bacteria that are photoautotrophs. The primary organisms that are photoautotrophs on this planet are plants. They're mostly plants that are photoautotrophs. But there are some bacterial species that can do it. There are a very rare number of bacterial species that are photoheterotrophs. They're still able to use sunlight for energy, but they're not able to use carbon dioxide as their carbon source. They have to use organic molecules as their carbon source to be able to make food. So they use sunlight for the energy source to make food, but they don't take in CO2. They take in other organic molecules, break those down, steal the carbon out of them, use that carbon to string together to make sugar that they can then eat. Not very many species of photoheterotrophs, not very many species of chemoautotrophs. These are ones that are able to make their own food, but they can't use sunlight to do it. They use the energy trapped in chemical bonds, usually sulfur compounds. Again, there are not very many species of chemoautotrophs. They break down sulfur compounds and get the energy stored in those chemical bonds to add carbon dioxide together to make sugar, to make food, that they can then eat for energy. Several species of photoautotrophs, not many species of photoheterotrophs, not many species of chemoautotrophs, lots of species of chemoheterotrophs. They are not able to make their own food. Oh, hetero means different. So different feeder, meaning they've got to eat other things to be able to get energy. They have to get their energy from chemicals. They have to get their carbon source from chemicals to eat things. You are a chemoheterotroph. Plants are photoautotrophs. Fungi are chemoheterotrophs. Most bacterial species are chemoheterotrophs. Most living things are chemoheterotrophs. This graphic shows the breakdown of how these different life strategies work. If it is an autotroph, it's going to make its own food. So chemoautotrophs and photoautotrophs make their own food. They'll use carbon dioxide as their carbon source. A photoautotroph will use sunlight as the energy source. A chemoautotroph will use organic chemicals as the energy source. So energy plus carbon means food. If you're a heterotroph, it means you use a different organic molecule as your carbon source. A photoheterotroph uses sunlight as the energy source. A chemoheterotroph uses organic compounds. Again, you and I, we are chemoheterotrophs, so our energy comes from other compounds and our carbon source comes from other compounds.